So, today we will look at some of the signaling pathways which are important in cancer. In the sense, there will be some aberrations in these signaling pathways. So, I said important signaling pathways that have a role in cancers is the title, because there are so many signaling pathways, hundreds of them. So, we cannot be studying every one of that. Broadly, they can be classified into oncogenic signaling pathways and tumor suppressive signaling pathways. So, we will study some examples in each of these, although I have not categorized that this way, but that is how we have to understand that. The tumor suppressive pathways already we have told, we have discussed, like P53 signaling, RB signaling and so on. Then TGF beta or so called SMAD signaling is also a tumor suppressive signaling pathway. There are other pathways which are listed here. So, you can see PI3 kinase, AKT pathway, MAP kinase pathways, Wnt beta catenin signaling pathway and cytokine signaling. Now, we will look at one by one. This is so called TGF beta signaling. What is TGF beta? TGF beta is actually transforming growth factor beta. When it was discovered, it was actually giving a stimulatory signal for cell proliferation, and so this was classified as transforming growth factor alpha, beta and so on. But later in most of the systems, this was giving a suppressive signal to growth, that is, it can suppress growth. That is why it falls under tumor suppressive signaling pathway. But the name stays, that is how when the discovery they made something, and so the name stays. This is not a single factor, it is a family of factors. And the most important one is obviously TGF beta. There are other family members, as many as 30 family members are known. They are basically proteins. So, there are many of them known. Some important members of the TGF beta family are listed here. The so called bone morphogenic protein, decapentaplagic, Mullerian inhibitory substance, inhibin or activin. So, these are some of the important TGF beta family members. They can act as ligands to various receptors. As I told you, their main function is to inhibit growth, controlling the growth and differentiation of epithelial cells. These ligands generally do that by induction of CDK inhibitor P15 Inc4B. CDK inhibitor, you know what is the function, cyclin dependent kinase. Since it is inhibitor, it inhibits growth, cell cycle. So, thereby there will be inhibition of cell growth. The other way of doing it is to suppress or repress the oncogene c-MYC. Actually, they will do both. TGF beta mainly functions through the induction of the transcription of CDK inhibitor P15 Inc4 and by repression of c-MYC. So, this is the mechanism. Now, we will look at it in some more detail. What is the receptor? There are two receptors for TGF beta, receptor 1 and receptor 2. Both are serine threonine kinases. Do not confuse with EGF receptor, which was a receptor tyrosine kinase, whereas this is a serine threonine kinase. The receptor 1 is about 53 kilodalton, receptor 2 is about 75 kilodalton. Interestingly, it is the receptor 2 which is constitutively active. I have repeatedly told what is meant by constitutive activation. Even without the ligand, it will be in the phosphorylated state, activated state. Actually, the receptor 2, although it is phosphorylated, it won't phosphorylate receptor 1 without ligand. So, it will the first the ligand interestingly binds receptor 2, then receptor 2 phosphorylates receptor 1. This is the important mechanism which has been unraveled in this case. So, you have to understand that. Otherwise, it is a constitutively active serine threonine kinase. Which one? The type 2 receptor. So, these facts have to be understood before we go into the details. So, the dimeric ligand binds to, first it binds to type 2. Dimeric means two molecules of TGF beta. They bind to receptor 2. This activates it and then it binds to receptor 1. So, they form a dimer. In this case, it is a heterodimer. You can see that. So, actually there will be a tetrameric complex because 2 ligands are there, 2 receptors are there. So, it forms a and not only that, it is the receptor itself is a tetramer because 2 copies of each of receptor 1 and receptor 2 are binding. So, the tetramer actually the receptor, ligand will be 2, dimeric ligand, but receptor, because each one binds to one receptor complex, and so you will have 4 receptors, in the sense 2 type 1 receptors and 2 type 2 receptors and 2 ligands. So, that way it is called a tetrameric complex with reference to the receptor, and it is a heterodimer, in this case oligomer, because so many things are binding. So, hetero-oligomerization takes place. This leads to trans-phosphorylation, as I have already told you what is trans-phosphorylation. Here in this case it is serine threonine residues in the cytoplasmic region of TGF beta R1. Remember, the R1 gets phosphorylated by R2. So, that has to be understood. Now, this leads to a series of interactions, what are known as signaling intermediates. These names are all derived from mostly Drosophila and other organisms. They might have discovered some genes, and that is how these names are given. SMA and MAD, a combination is called SMAD. SMA is one gene, MAD is another gene. A combination of that is called a SMAD actually. These SMAD genes, SMAD proteins are the signaling intermediates, very important signaling intermediates. Again, like TGF beta, there are many SMADs known, 1, 2, 3, 5, 4, 5, 6, 7, 8 and so on. There are many of them, but we can classify them into 3 types. Regulatory SMADs, which are usually 1, 2, 3, 5 and 8, or they are also called regulatory SMADs is enough, R SMADs, otherwise they are called R SMADs, it is a short form. Co-SMADs, as they are called. One example here is SMAD4. Generally, the co-SMAD, SMAD4 is not getting phosphorylated when the signal transduction takes place, it only binds. But there are also, as I always told you, whenever there are activators, there are inhibitors. So, this phosphorylation can be inhibited by certain types of SMADs called inhibitory SMADs or so called I SMADs, SMAD 6 and 7. So, basically these SMADs, the signaling intermediates, are transcription factors that are generally getting activated. They get phosphorylated, except for SMAD4, other things are getting phosphorylated, and they have different domains, what is called MH1. M means this is a SMAD, I told you, is made up of SMA and MAD. So, this MH1 means mad homology domain. That is why it is called MH1 and MH2. It has got homologous sequences to the MAD gene. So, that is the idea. These names I told you, decapentaplegic, all these names, they have been derived from Drosophila and other organisms. So, the MH1 domain is very important for DNA binding. MH2 domain is important for oligomerization and receptor interaction. So, this is the important thing that you may have to understand. Now, let us look at the signaling pathway as such. Now, we have just described the various partners. Now, we will see how the signal transduction takes place. So, TGF beta is there, it goes and binds to receptor 2, actually 2 molecules will bind. So, there will be 2 receptors R2 receptors, 2 R2 receptors, and then this will form a tetrameric complex, tetramer with reference to the receptor, because there are 4 receptors, 2 R2 receptors and 2 R1 receptors. I hope it is not confusing. So, that forms a complex along with the ligands, and this results in the phosphorylation of R1, and that recruits, this phosphorylation is the activating signal for the recruitment of the regulatory SMADs. TGF beta 1 is the famous example. For that, the SMADs will be 2 and 3. SMAD 2 and 3 get phosphorylated, because each one, I told you there are 30 members of the family. So, they may engage other SMADs. So, do not worry about that, because we cannot be remembering all of them. So, just remember TGF beta 1, and once it binds the receptors, then SMAD 2 and 3 are getting phosphorylated, and this will form a complex with the SMAD 4, which is generally not phosphorylated. So, all of them form a complex, SMAD 2, 3 and 4, phosphorylated SMAD 2, phosphorylated SMAD 3, unphosphorylated SMAD 4, they form a complex. It is this complex that binds, because I told you these are transcription factors. So, what is the role of a transcription factor? It has to enter the nucleus from the cytoplasm. Now, signals are carried to the nucleus and bind a specific DNA sequence, which we call as SMAD binding element, SBE, SMAD binding element. It binds there. It is not only these SMADs are going and binding, but there will be a transactivation complex. A number of members will be there in the transcriptional complex. They are called co-activators or repressors. Both will be there. It is always a balancing force. There will be some repressors, there will be some activators. Ultimately, if the signals are strong enough, then it will lead to activation. If the repressors are strong under some situations, then the signaling will not go. For example, SMAD 6 and SMAD 7, if they are there, they will block. Because usually SMAD 6 and SMAD 7 will block even at the earlier stage. SMAD 2, 3 phosphorylation can be blocked by SMAD 7 and SMAD 6. So, even at an earlier step they can block. So, there are other repressors. So, this results in transcription of genes which are involved in growth inhibition. Like I told you, one of the famous example is that CDK molecules, CDK inhibitors. Now, you may wonder why we are studying this, because we are studying this because in many cancers, especially in colon cancer, almost 30%, 1/3 of tumors have mutated TGF beta receptors. The remaining cancers may have mutations in other partners. In the signaling intermediate, there will be so many intermediates, and some of them may get inactivated. in colon cancer. In other cancers also some people have found, even we have reported in cervical cancer, SMAD2 and 4, some loss of expression in cervical cancer. So, in many cancers there can be, because these are having tumor suppressive role, and I told you in cancers tumor suppressive genes will be either mutated or deleted. Oncogenes will be activated, tumor suppressor genes will be inactivated in cancers. So, that is there, but that inactivation may happen through several mechanisms. Mutation is not the only one. Later on I will talk about epigenetic alterations which can also result in loss of expression. It need not be mutation. Net result is inactivation. That inactivation can be through mutation, deletion kind of mutations, or through epigenetic alterations. Now, cancer cells specifically become insensitive to TGF beta. I told you TGF beta is a growth suppressor, but cancer cells will not obey this, which means, what it means is, since they have mutations in the receptor, the signaling may not take place. So, the suppression is not there. If there is no inhibition, there will be a conducive atmosphere for growth. So, that is a simple idea you can have. The cancer cells become insensitive to TGF beta due to loss of expression and mutations of SMAD proteins. So, that is the idea. Now, that is about TGF beta signaling pathway. You can also look at some of our publications. We had a few publications on TGF beta. Just as a matter of interest, you can go through them, and we can, if you have some doubts also, you can ask. The next pathway, this is oncogenic actually, PI3 kinase or so called AKT pathway. This is a very important pathway. I told you what is inositol. That is a cyclohexane, all the 6 carbons are hydroxylated. If you put a OH group in all the 6 carbons, that becomes inositol. This is one important signaling molecule, because inositol can be phosphorylated because it contains OH groups in all the carbons. So, it can be phosphorylated at positions 3, 4, 5. So, if it that is called phosphatidyl inositol derivative at the 3 position. PI3 kinase is an enzyme which catalyzes the conversion of the diphosphate 4,5 diphosphate into 3,4,5 triphosphate, phosphatidyl inositol 3,4,5 triphosphate. So, that is the idea. What happens is, see, kinase phosphorylates. So, if there is a diphosphate, it adds one phosphate group and that becomes triphosphate. The so called phosphatidyl inositol triphosphate, which we call PIP3, is a very good second messenger. You all know what is second messenger and first messenger. First messenger is the ligand. The one that takes the signal from the first messenger is called second messenger. So, this is one example. Other famous example for second messenger will be cyclic AMP. Cyclic AMP is the most well known second messenger, not for this pathway, but for other pathways, but it is a very well known second messenger, and this PIP3 is also a second messenger. They are involved in cell growth control naturally, chemotaxis and glycogen synthesis also, apart from their role in cancer. Now, the PI3 kinase is activated by several pathways. I have just listed 3 of them. Interaction with activated receptor tyrosine kinases. Receptor tyrosine kinases we already studied. For example, there can be growth factors, hormones like insulin. They bind with their receptor tyrosine kinase, and one of the signaling intermediates is PI3 kinase. It can, for example, through RAS also, RAS is an oncogene, it can activate PI3 kinase. There are many members, later on we will look at it. This is the PI3 kinase catalyzes this reaction, phosphatidylinositol diphosphate 4,5 diphosphate is phosphorylated to become phosphatidylinositol 3,4,5 triphosphate. So, that is the reaction PI3 kinase catalyzes. Not only that, activation through GPCR, you remember G protein coupled receptors. So, they can be activated through that also, their own receptors can get activated and they also activate PI3 kinase. And these are the three things. What are the three pathways? One is through receptor tyrosine kinase, the other one is through RAS pathway, the other one is through GPCR. So, three pathways can be responsible for the activation. Now, this PI3 product, the so called PIP3, this conversion is as you can see is a reversible reaction, but it can be inactivated by another molecule called P10. P10 is a what kind of molecule it is? If this is phosphorylation and if it is reversed by this, it is a phosphatase. P10 is a phosphatase. Because PI3, that PIP3 is oncogenic and now that is activated through phosphorylation. You see that from diphosphate to triphosphate, it is phosphorylation. That we are calling it as an activation mediated by PI3 kinase. PI3 kinase phosphorylates, but P10 dephosphorylates. So, it is a phosphatase and so it acts as a tumor suppressor because PI3 kinase is oncogenic and now this P10 inhibits its activity and so it is a tumor suppressor. That is very important because it is a well known tumor suppressor that negates the effect of PI3 kinase because it inactivates PI3 kinase in a way. Not exactly inactivates, it nullifies the effect of PI3 kinase because by dephosphorylation whatever PI3 kinase has done is undone by P10. So, that is what has to be understood. P10 basically promotes the conversion of triphosphate to diphosphate because it is a phosphatase. So, that has to be understood. These are important because PI3 kinase is very important in cancers. P10 mutation is also important because the tumor suppressor here is an example where oncogene is activated, the tumor suppressor gene is inactivated. P10 mutations are there. There is another kinase which is called AKT or protein kinase B, is actually activated by binding of PIP3 to the pH domain, plextrin homology domain, directly or indirectly involved in cell proliferation, and it also helps in preventing apoptosis because for oncogenesis to happen, the cancer cells want to prevent apoptosis because they do not want to die after all. So, that is prevented. These AKT kinase also mediates the effect of metabolic effects of insulin like glucose transport, lipid metabolism. These are all functions of insulin, glycogen synthesis, protein synthesis and so on. For our convenience we are studying these things, but then they will have other actions also naturally. Now, AKT and apoptosis, about apoptosis I am going to take a separate class. Apoptosis is, all of you know, is a programmed cell death, is akin to cellular suicide, you can say, because the cells decide themselves to die under certain conditions. We will look at those conditions and all that. Here I am just giving an introduction because we are talking about AKT and how it is able to regulate apoptosis. So, the receptor tyrosine kinases, in their signaling pathway, PI3 kinase is a signaling intermediate, and that can also activate PKB or AKT, and AKT controls a number of molecules which are involved in apoptosis. What are the molecules? Later on we will study about these molecules. Just understand that many of them are involved in apoptosis. BAD is another gene, it is a pro-apoptotic. Caspase 9 is again pro-apoptotic. IKK means the it is the NF kappa B activator kinase, it is a kinase. I kappa B kinase. IKK means I kappa B kinase. There are many of them. Here I have given a general name, I kappa B kinase. And then MDM2, which is an inhibitor of what? P53. CRB and GSK3. GSK is nothing but glycogen synthetase kinase, GSK3. And then through BAD, for example, in this case it kind of this AKT blocks BAD. BAD is pro-apoptotic. So, when you block that, apoptosis is prevented, right. Similarly, caspase 9, when it is inhibited, you are regulating the apoptosis negatively.
Of course, there will be other roles. ICA kinase inhibition will actually block NF kappa B, which will induce apoptosis in that case, because NF kappa B is anti-apoptotic. So, it will generally, you can say, it can regulate apoptosis, because it can do both. Sometimes
both signals, apoptotic as well as anti-apoptotic signals, but mostly anti-apoptotic signals. That is what happens. For example, P53 gets blocked because it activates MDM2. So, if it regulates NF kappa B, the caspase cascade, the BCL2 formation and the P53 inactivation, all these events eventually can regulate apoptosis. We will later on study about apoptosis, what are the different apoptotic signaling pathways, how they can be regulated and all that. For the time being, just understand AKT is oncogenic and it does that by regulating apoptosis. In other words, kind of negatively regulating apoptosis, and it regulates a number of molecules involved in apoptosis, which are listed here, like BAD, caspase 9, IKK, MDM2, CRB, GSK3 and so on. A number of genes are controlled by AKT. Now, I told you that P10 is inactive by mutation. Loss of function mutations are common in colorectal cancers. P10 is its activity is lost in colorectal cancers. Activating mutations, similarly PI3 kinase gets activated, or AKT also. Activating mutations usually lead to constitutive activation of this pathway in many cancers. Numerous components of the PI3 AKT pathway are targeted by, there are other mechanisms like I told you, amplification, mutation, translocation. So, this is a very important one, only its importance is probably superseded by RB, retinoblastoma and P53, because if you take the tumor suppressive pathways, RB and P53 play a very important role. because I told you how P53, more than 50% of the cancers you have P53 mutation, right. So, that way they are very important, RB and P53. Apart from that, this mutation of the AKT pathway also is very important. If you can inactivate P53 or AKT, then also you can it can result in inactivation. But then P10 loss and activation of PI3 is what is happening in the cancer. Do not get confused. What I meant is activating mutations are common in cancer, mutations of PI3 kinase common, and similarly loss of function of P10 is also common in many cancers. So, these mechanisms are very important in with reference to cancer. The next pathway that we are going to discuss is very important. It is often called RAS MAP kinase pathway. Mitogen activated protein kinase is what we call as MAP kinase. Mitogen activated protein kinase. They are usually activated in response to growth inducing signals like insulin, other growth factors, EGF, all those kind of things. There are many MAP kinase pathways, because there are many MAP kinases. They are also known as ERKs, means extracellular regulated kinase. ERK1 and ERK2 are also MAP kinases. The MAP kinase, another name is ERK1, ERK2. So, extracellularly regulated kinase, that is what is ERK full form. C-Jun N-terminal kinases, we call them as junk or JNK or whatever, JNK 1, 2, 3, there are many. See, many of these molecules will have substitutes. This is an important concept also in biology for you to understand. Whenever there is a molecule and it does some function, there will be some substitutes. Of course, with slightly less efficiency or with more efficiency, it depends. One may take over the other, like we do for a spare bus or something like that. So, substitute basically. The substitution can be fully substituted or partly substituted, something like that. So, there will be some such similar molecules like JNK 1, 2, 3, structural analogues you can say, and so on. P38 kinases, again as I told you, ERK1 and ERK2, there are ERK3 and ERK4 and ERK5 also. There are many MAP kinases, that is what it is. In the next slide probably I will just show you that. This is often called kinase cascade. MAP kinase is phosphorylated by what is called MAP kinase. And that is phosphorylated by MAP kinase. So, this is how the names are given. You can see that. See here, you go from the bottom. MAP kinase we know, and that is phosphorylated by MAP kinase. You can see that, and that is phosphorylated by MAP kinase. It will be slightly difficult to understand, but phosphorylation is the activating signal. These kinases also they phosphorylate. They are also activated by phosphorylation. So, there should not be any confusion regarding that. The enzyme also gets activated by phosphorylation, and its job also is to phosphorylate. So, you have to understand that. So, these cascades are very important. I have written, recall what was said about signaling cascades. What it means is that, what I try to say is that they are helpful in amplifying the signals, because they are spreading it to other molecules. They can phosphorylate different molecules, and that way the signal gets diversified as well as amplified. I will come to that amplification. That is a very important thing. You remember I told you about scaffold protein. So, it can basically it can interact with many number of proteins. One protein interacting with different proteins will be an ideal one for being a scaffold protein. So, that is the idea. You can see that these three kinases form something like a scaffold, and the signals can come from different molecules. You can see here the signals can be received by from growth factors, stress, UV radiation or heat, hormones. Here itself you can find how the signals differ in their characteristics, because heat is also a signal, stress is also a signal. You cannot put them under one category, protein or lipid, something like that. It's not like that, because some are radiation, heat, temperature, anything can be a signal, ions can be a signal. So, ultimately these lead to some biochemical effect. In this case it is mostly proliferation. What we want to understand from this is the scaffold function of these proteins. This is very important in understanding this. This is really the so called MAP kinase pathway. A typical example is epidermal growth factor. See here EGF, it binds to its receptor, usually a dimer. It is not shown here that way, but it is usually a dimer, because it is a receptor tyrosine kinase, and that binds to GRAP2. The moment it is getting phosphorylated, GRAP2 is an adapter protein. If you remember that SH2 domain and all that I have discussed, and that will immediately recruit SOS. These names are very funny. SOS means son of 7 less. There are some 7 receptors and things like that from Drosophila again. You will find all these son of 7 less, daughter of pentaplegic, something like that. All these kind of names will be there. Anyway, let us not worry about it. So, this is known as SOS. This gene is also important. Then that recruits RAS. Now, the RAS from RAS has a GTPase activity. So, it can be converted to GTP RAS, which will again phosphorylate RAF. RAF is another molecule, and then it is connected to the MAP kinase, which is also called MEK. MEK is nothing but MAP kinase, and that naturally phosphorylates MAP kinase, and that MAP kinase can phosphorylate different molecules. Here you see the diversification exactly in this place. If you can note that. Here I was showing something like this is sometimes called vertical signaling, and here you see it is spreading, because MAP kinase phosphorylates here in this case MYC, and then there is another called ribosomal S6 kinase, RSK it is called, is getting phosphorylated. See here S6 subunit is getting phosphorylated, that is why it is called ribosomal S6 kinase, something like that. So, the S6 factor is getting phosphorylated, and then there is one M and K getting phosphorylated. So, 3 things, 3 molecules M and K, RSK, MYC can be phosphorylated, but all of them can ultimately result in the transcription of certain genes which will be helpful in proliferation. First, scrub is another mediator. M and K phosphorylate scrub, and that can result in the transcription of genes which are ultimately involved in proliferation. So, basically this RAS MAP kinase pathway is involved in cellular proliferation. So, this is how growth signals are emanating from different growth factors, and they ultimately they are able to stimulate growth. Basically, they will promote cell cycle. See, they will stimulate cell cycle. so that more divisions will take place.